Hi everyone, let's talk about clinical trials today. Clinical trials are considered the strongest single study design. And there's several different ways that people say clinical trials. Sometimes it's controlled clinical trial, or controlled trial, randomized clinical trial, randomized controlled clinical trial, randomized controlled trial. In this class, we're going to use these phrases interchangeably, but really they do have very subtle differences. So a clinical trial is called that because it is an experiment where the investigator decides where the subjects should be assigned. And so subjects don't actually choose what group they want to be in, but rather they're assigned through a randomized process to get into the different groups. Because there's this randomization, which is a very strong way to allocate people to groups, it's considered the gold standard of study designs. Some people mix up the term randomization with random selection. Randomization deals with how people get into the groups. It's sometimes a shake of the dice. It might be using a random number table. It might be computer-generated randomization. But random selection instead deals with how people get into the study. So random selection is choosing people randomly to be in a study. So maybe I would choose 5 students out of the class to fill out a survey. But that doesn't mean that it's a clinical trial. A clinical trial comes when there's randomization into the different treatment groups. So here's what randomization looks like. We have an original group of subjects. Randomization occurs, and subjects go into one group or the other. Sometimes we stratify our randomization. So stratification occurs when first we decide we want to separate people into 2 different groups, and then they're randomized from there. For example, sometimes we might stratify into males and females, and then randomize from there. This would help prevent all of the males being in one group and the females being in the other group of treatments. And it allows us to have an even number of males and females in each of the different treatment groups. When we randomize, what we're doing is we're actually decreasing selection bias. And that selection bias can happen either by the investigator deciding, I think this subject would go best in that group, but this one's not doing so well, so let's move them in the placebo group. Or by the subject. Sometimes subjects want one treatment over the other. And when they receive that treatment, they're very happy and they feel as though they're doing much better. Randomization also evenly allocates based on baseline characteristics. And these are known and unknown. Usually when you look at Table 1 of any article, you'll be able to see the baseline characteristics, and you can determine if we say randomization worked. This is an example of Table 1 where we're looking at the baseline characteristics of age, BMI, and percent of people that ever smoked. When we look at the control and the intervention, what we see is that those 3 baseline characteristics are approximately the same. And our p-value is not statistically significant. This is what we would expect to see at the beginning of a study. When we randomize people into 2 different groups, we don't want those groups to be different on any characteristic. That's what randomization is all about. And so we expect to see those p-values to be more than 0.05, which indicates it is not statistically significant. Blinding is also a big deal in clinical trials. We can blind the subject so that they don't know which treatment group they're in. We can blind the clinician so that they're not aware of the different treatment groups. Sometimes the clinician is blinded through the use of a placebo pill. this medication may look exactly like the regular medication, the true intervention, but the clinician doesn't know which is which. Now, if both the subject and the clinician are blind, we call that double blinding. But sometimes the clinician has to know the different groups, and in which case it's hard to have double blinding. But nowadays, the investigators, the researchers, or the research assistants can also be blind. What happens with this is they will come in and do different outcome measures without knowing which group the patients are in that they're doing outcome measures for. And so we can still have double blinding even if the clinician within the study knows which treatment group their patients are in. Something that is beneficial within clinical trials when there is blinding is to ask the subjects, what group do you think you were in? This really helps us assess whether or not subjects are aware of which group they're in. If 100% of the placebo group knows that they were in the placebo group, well, then we need to be concerned about the results, because these people are going to be thinking that was fake, I didn't get a real treatment, which might actually affect their outcomes. We also need to ask the treatment group, what group do you think you're in? If 100% of the treatment group thinks they were in the placebo group, well, maybe that would kind of even things out if also the placebo people think they're in the placebo group. But it's very important to ask so that we're aware of, might there be a bias based on that factor. In clinical trials, we see this type of a flow chart. It's called a consort flow diagram. And what this shows us is how many people were assessed for eligibility within the study, and why were they excluded from the study. It then shows us how many people were randomized into each group, how many people received the treatment, how many didn't receive it, and why not. And then with follow-up, it shows us how many people completed all the way through to follow-up, and how many discontinued, and why did they discontinue. In clinical trials, we also see long-term follow-up graphs. Usually in a clinical trial, we would expect to see follow-up in the order of 1 to 2 years. And so it really depends on what the study was looking at, but with most studies looking at different therapies, we expect to see a long-term follow-up of at least a year. With the analysis of clinical trials, there's something called intention to treat analysis. This deals with how we analyze the data. Now, in clinical trials, we randomize people to different groups. Sometimes people don't get in the group that they want, and they might switch over to a different one. Or there might be a mistake in randomization where people ended up taking the wrong treatment, and the researcher was the one that caused that. Sometimes people are enrolled in a study and they weren't actually eligible for the study, but they continue through the study because they were randomized into it. All of these factors can really affect the baseline characteristics. If we start getting rid of the data points from the people who perhaps didn't continue in the treatment group to which they were allocated, well, once we start throwing all that data out, now our baseline characteristics in the two different groups might start being very different. And so the intention to treat analysis really is the purest way to do the initial analysis. Anyone who's randomized into a group gets analyzed in that group. Sometimes then, if there were a lot of factors that cause subjects to perhaps switch groups or do extra treatments, have different co-interventions, then the statistician may say, let's do a secondary analysis where we take all of those data points out for people who didn't receive the allocated treatment. Let's take them out and do a secondary analysis to see what the results have to say then. But when you read clinical trials, you should expect to see intention to treat analysis as the primary analysis. In clinical trials, we will sometimes see that there is some sort of an influence based on who funded the trial. And so this is very important to look at. If you see that a drug company, let's say, funded a trial and the results are very, very positive, but let's say NIH funded the same type of trial and the results are not as positive, sometimes there's suspicion that the drug companies are kind of boosting the results or somehow falsifying things. And I'm not trying to say that drug companies necessarily do this, but there are some studies looking at the influence of who funded the study on the results. And so there can be these types of issues. And so nowadays we don't expect that to happen as much as it happened perhaps in the past, because we do have such strong disclaimers. Anyone who's involved in a clinical trial must disclose if there are any potential conflicts of interest. Clinical trials have a lot of ethical issues. So besides the conflict of interest, we have to think about the patients. Sometimes these active treatments are brand new and they end up not being safe. And so in clinical trials, we expect that adverse events are always reported. And if there are no adverse events, researchers should report that as well. In clinical trials, investigators might withhold treatments from a certain group. So one group might get placebo care, and withholding that treatment, that might be an ethical dilemma. And so number 3, there are a lot of issues surrounding placebos. Is it really ethical to tell someone this is a treatment when in fact it's not? Clinical trials also have informed consent and reconsenting. When a patient enters into a clinical trial, they sign a form. First they're told about the study, then they have to kind of repeat back what the study's about and answer a few questions. But that's not the end of it. These days in clinical trials, patients are supposed to be reconsented at certain time points to make sure that they remember what the study is about and that they continue to agree to go through that type of study design. Finally, with clinical trials, there's usually stopping rules that are overseen by a data safety and monitoring board. A DSMB is charged with looking at the data, sometimes ahead of the end of the study, just to make sure that one group isn't getting very, very sick from the new treatment, or that another group isn't getting very, very well. And on either of these ends of the spectrum, the DSMB may decide we need to stop this trial because it's unethical to continue, either because too many people are getting ill or dying, or it's not fair to those not receiving, the treatment in one of the groups because everybody's getting better and we need to stop so that everybody can receive this treatment. So the good thing about a clinical trial is that it's the gold standard for evaluating different types of treatments. Randomization can decrease bias, as can blinding. And when we look at clinical trials, we usually see very structured therapies so that the investigator, the researcher, can fully describe very succinctly in the research article as to what happened to those patients. And because of all of this control, it's considered the strongest study design. However, all of this structure isn't really what clinical practice looks like, necessarily. And so when a clinician might give a certain amount of drug or medication to one patient, they might give only half of that to another patient. And so really, sometimes clinical trials aren't generalizable. We can't always apply the results of a clinical trial to all of our patients. Sometimes it's unethical to perform There are people who talk about placebos as we really shouldn't have them anymore in science. There's also some studies where we can't randomize people into one group or the other. For example, if we think about clinical trials, let's say, that lead to disease, we can't say I'm going to randomize one group to smoke 2 packs of cigarettes a day and the other group isn't going to smoke at all. That would be unethical. So clinical trials are very large and they can take a lot of people and can be very expensive. And so this can be a disadvantage as well. So if you have any questions about clinical trials, please feel free to let me know. I'm happy to talk about it.